So again, as I say, there are different types, but the useful one is type 2, so I'm jumping into restriction enzyme type 2. What are their characteristics? They have separate subunits, but the key important restriction sites, typically, I already said that, palindromic, cleavage at defined position, and they create these discrete restriction fragments. So they are very useful for a targeted restriction digestion purposes, for your purpose, for various purposes. If I am using pulsed field gel electrophoresis, like Riley will cover this afternoon, I would select for a rare cutter enzyme that cuts the genome of that organism, and I'm not going to the detail of that, but then you can predict in how many places that enzyme would cut the organism, separate it, and you're looking at the macro restriction profiling. That is one genotyping method. Or you might be using EcoR1 to cut specifically a gene locus and then clone it into a plasmid vector fragment, transfected for various purposes. So they are useful, applied for various purposes. One advantage of type 2 is they do not need ATP energy to work. It's very easy, just you mix whatever ingredient you need, the enzyme, your DNA, some certain temperatures they are active, 37 overnight or couple of hours, you digest it. No need for energy. Okay. So typically, there are two different types of cutting they use. One is with overhang, or also called sticky ends. That's the most common one. And some of them with the blunt ends. We'll look at that as well. So if you look at, again, no animation here, the GAATTC of EcoR1. So GAATTC, GAATTC, it cuts, with a specific defined sequence, right? Am I correct, by the way? Yeah, right after the G, it cuts, okay? So you have this. So what happens is, if you digest this and then you have this cut, so you can really have this overhangs. The double stranded will continue whatever bases are before and after. It can be ATT or any basis, but you will have this, the end of it will have, a sticky end is called overhang, a single strand part of the end of the digest. Is that clear? Because that's very important, because you want to digest and then maybe you want to attach, that's why we call it genetic engineering, right? So you want to move one gene, attach it to this digested vector or other purpose. So the sticky end is very important in preparation for your reaction. If everybody on the same page, clear? Okay? I think somebody has a question. Okay, no? Okay, so here is an example of a sticky end. EcoR1, I just showed you, GAATTC. It digests it, not just in symmetrical blunt end, but with some overhang, single strands. Look at here. Okay, so it digests right here. So you have this yellow and the red part separating. So my gene target is here. For example, I want to clone MAC-A gene into a vector, clone it for sequencing, other purposes, or various purposes. Less so, we don't use much in epidemiology, by the way, but in virology, or you are doing some pathogenesis work. And then I digest the genome of this organism, in this case MRSA. Let's say I have an MRSA from patients. I want to know, sequence, and try to understand whether it is the same MAC-A gene circulating in the population. Just let's say that's a simple question. This is MAC-A gene, I digest it, then I digest the plasmid vector. Okay, this is, let's say, PAC18 or whatever, that a vector, a carrier of my gene to clone. So I digest it using the same enzyme or a different? It's a simple question, but just to make sure you are alert enough. Same enzyme, you have no choice. I use EcoR1 to digest the packaging. I use EcoR1 to digest my genome fragment. So I look at, let's say in this case there is a make in between, you attach that fragment. Okay, so with your vector. So here it is attached one. And then you can play with it. You can do many things. You can transfect it into E. coli, create a library, sequence it, whatever you do. But the point I want to make is restriction enzymes are important because you know what is at the end of that fragment. Because you are using EcoR1, you know what EcoR1 recognizes. So at the end, the fragment, that sticky overhang, would have this GAATT on both sides. Then you can use the same enzyme on your vector and attach it. That's one application, but you can use it for various purposes. Look at that in the mass sequencing purpose. The other types of type 2 enzymes are with no sticky ends, no overhangs. They have blunt end. Like SMA1, it recognizes CCC, GGG, the same again, palindromic, right? You have read it's the same. And it cuts right in the middle. There are no overhangs. Okay? SMA1 is, in fact, that's the most common enzyme we use for PFG of MRSA. Okay, to look at the clonality of MRSA, if you are using PFG, SMA1 works well. There are some livestock-associated MRSAs these days. You can't use just a straight SMA1. That's a little bit more complicated, but we can maybe address later on. But you have to have isoschizomer of SMA1. Okay, XMA1 or CFR, some other enzymes to be able, that's a different issue, but just to give you an example, SMA1 with the blunt end is one of the good examples that's commonly used restriction enzyme. Okay, clear? So now, how frequently do these enzymes cut? How would I know? How frequently do they cut? Just think about a genome, whatever organism you work on, Salmonella 4.5, mega base pairs. I keep saying megabytes, so I don't know, for some reason that gigabyte is confusing with base pairs. So mega base pairs, or Campylobacter 1.4 mega base pairs, or whatever organism you use. And you have this enzyme, you know the recognition sequence. You know the ATGC proportions of your organism. I don't know what your organism you work on, but you can have a good estimate of how many fragments can you get, right? But what factors do that govern what's the frequency of cutting? One is clearly the length of the recognition sequence, correct? Do you agree? How? Why is that important? One word, statistics word, not even life science word. The probability, right? If your enzyme is recognizing a short sequence, what is the probability of finding that short sequence? High or low? High. If it is recognized longer sequence, low. So it will be a rare cutter. Frequent cutter, rare cutter. Okay. So that is one way very important one. Let's do this exercise. If you have a 50 kb DNA genome and you are using a 4 cutter, 4 frequent cutter that recognizes 4 sequence like MSC1, AATT, how many fragments do you get? This cannot be a bacteria, but let's say it's a virus, 50 kb. Okay. You have a 4 cutter, AATT. How many fragments do you get on average? Okay, let me qualify that. On average, definitely, okay, because we don't know the composition. Can you calculate?
10.
Okay, good. Keep doing. Anybody here? Calculating. How many fragments on average would you get?
256.
Okay, 200, I'm liking it. On average, so 200? 56, okay. Agree? Somebody who's fast calculator, that's great. So you guys are... We're close, but I don't quite agree, but that must be. Okay, what did you do? So okay, you're trusting your friend. That's a smart friend. That's good. Okay, so what do you do? How do you come up with that? But that's, we're close, so let's see. Okay. How do you calculate it? Here is what I did, but I think you did great, because look at this 256. That's really smart. You don't get 256 fragments. Just wait. It should cut once every 256. Excellent, because the 4 positions has a probability 1/4, right? Times 1/4, times 1/4, times 1/4. The chance of finding those 4 together is 1 in 256. So how many fragments do I get? So that's the first important step. But there is one more step. You have to consider the genome size. In this case, I told you 50 kb, 50,000. Don't you need to do one more step? But that's excellent. Okay, it cuts 1 every 256 on average. On average, it cuts 1 in 256. That's excellent. But I have 50 kb. Would it be the same as if I have 4.5 megabase pair? 4.5 million versus 50,000?
195.
Excellent. That's collaboration. That's great collaboration here. You see, that's why we all collaborate. So, exactly. So you get on average 195 fragments. Thank you very much. Is everybody clear? So you can estimate, based on what is the recognition sequence, how many fragments do I expect to find? Okay, it cuts 1 every 256. The genome size of the organism I'm working is 50 kb in this case. 50,000 divided by 256, 195 on average. Okay? This is assuming random or distribution of the 4 bases. Everybody clear? But this is important thing as you develop. We had a situation about, I think, 8 years ago. There is one marine biologist came to my lab and asked, this is a brand new organism, we're finding it from deep in the ocean, very interesting story. But they wanted to develop a PFGE method. Okay, where do you start? But you have to design, first of all, they don't know the genome, but there is lots of trial and errors, but at least we have to have, they had some estimates of the genome size, I don't know, but there was no full genome sequence. Some estimates, so we had to consider that size and maybe try this enzyme, a rare cutter to get, but I know they published it in some good journal after, and they were able to develop a PFGE method, but the key thing I want to say is, you have to have those information, your recognition sequence, the length, and then the genome size to be able to predict how many fragments do you get for your genome. And you may have different applications. Okay, now just to wrap up restriction enzymes, if you go to New England Biolabs, I think Daniel Nathans and his team established that unit. That's excellent resource. We all order our restriction enzymes from New England Biolabs, and there are lots of detailed resources in terms of what enzyme recognizes what, how long, and all kinds of that information. Okay? Clear? Claro? Here is the website information, okay? So just wrap up restriction enzyme in terms of application. Again, you will, starting from this afternoon, you will see a lot of utilities utilizing restriction enzymes, but various purposes, cloning, gene mapping, of course, sequencing purpose we'll look at soon in the next lecture as well. Restriction, fragment length, polymorphism, PFGE, a number of other genotyping methods that we use these restriction enzymes for. Okay, use them for toxinotyping of Clostridium difficile and many purposes. Very useful tools as a molecular biology tools, but for epidemiology as well as other purposes. Okay. Any question on restriction enzymes? Then we'll move into sequencing, and that way we'll wrap up the core methods.